Hi, everyone. My name is Joanne Mudge. I work at the National Center for Genome Resources, or NCGR, in Santa Fe, New Mexico. I'm here to tell you about our research. We apply machine learning approaches to pangenomic graphs in order to connect genotype to phenotype. And I'd like to thank the organizers for making this happen, as well as my colleagues, especially Buwani and Indica, who really helped put this poster together. So to start off with, a pangenome is simply a collection of genomic information from different individuals that are related to each other. And we work at a nucleotide level, so instead of just looking at genes, we look at every nucleotide in the genome. We are comparing to genome-wide association studies, or GWAS, which has a proven track record for connecting genotype and phenotype. This is a reference-based approach where you take one reference and then align all the other individuals back to the reference, and so it introduces a little bit of bias. And so we are proposing a reference-free approach, which we call PWAS, Pangenome-Wide Association Studies, because we use a pangenomic reference. And this pangenomic graph is a reference-free approach where there's not one individual who is set as the reference, but all individuals have equal weight. And the pangenomic graph contains all of the genetic variation that are contained within each of those genome assemblies. We go through the graph and find conserved regions, which we call frequented regions. And then these regions we can use as features in machine learning models in order to create genotype and phenotype relationships. So a frequented region, or FR, is a hotspot in a pangenomic graph. In this case, we use a CDBG, or a compressed de Bruijn graph. And these hotspots regions are co-visited by a set of supporting paths from individual sequences in the pangenome. And so the paths are just the paths that each of those individual genome assemblies takes through the graph. And so when you have a set of co-visited paths, you just have individuals that are similar to each other. I'm not going to go too much into the details here, but I do want to pull out a couple of the parameters. So the K parameter, or the Kmer, is the length of the string that you're using in order to build up the de Bruijn graph. The alpha is your penetrance parameter. And so as you're looking for shared paths within the graph by different individuals, you can set this so they don't have to be exact. You can allow a few nodes here and there, a small fraction of nodes, to be different between the individuals that are sharing that FR path. And you can dial this up and down in order to look at kind of the high level view of how things are related and getting kind of the broad synteny, or getting down into the details and how exactly, and looking at every difference there. The kappa parameter is our maximum insertion parameter, looking at the maximum number of nodes that are outside of the FR path. And so this is looking at, if you have a bunch of genomes that are sharing the same paths, and so you're putting them into the same FR, if one of them then has an insertion, but otherwise it's exactly the same as the other ones, do you want to keep that into the same FR path or not? And you can tweak the kappa parameter in order to make that distinction. We have two yeast data sets. These are from two different studies. So one has 100 yeast strains and 49 phenotypes, and one has 200 yeast strains and 35 phenotypes. Most of these phenotypes are how well yeast is growing on different media. We are comparing back to a baseline of GWAS, genome-wide association studies. So in these studies, they're scanning for common variants across the population, and then they're associating these variants with different phenotypes in order to figure out which regions of a genome are responsible for differences in phenotype. We are comparing this to our machine learning models. And so we actually have two models that we're working with in order to be able to better understand the processes going on. And so one is the FRML, where we're using FRs as a feature and comparing those to phenotypes. And then the other is SNPs-ML, where we're using exactly the same SNPs that are used in the GWAS study and using our machine learning method. And that helps us to sort out whether it's the FRs or the SNPs versus the SNPs, or the ML versus the statistical genetics methods that's really making any differences that we see. Our classifier is a random forest, which tends to work well on genomic data, which can be very messy. So you can see in this picture in the middle here, the 3 tracks that we're using, the GWAS, the FRML, and the SNPs-ML. And so the GWAS starts with SNPs and uses statistical genetic methods to connect to phenotype. And the SNPs-ML also starts with SNPs, but as features in a machine learning regression, and the FRs uses those frequented regions as features in the machine learning regression. So here's our experimental setup. We are using the scikit-learn Python library in order to implement our machine learning models. We use a 5-fold semi-nested cross-validation in order to train and test and also validate our model. And we are using a performance metric of root mean squared error, or RMSE. And essentially what this tells you is how different your predicted phenotypes are from the actual phenotype. And so for each of those individuals. And so you want to minimize this. You want this to be as low as possible, because that means that your predictions are very close to the actual phenotypes. This table down below shows some of the results. So RMSE for the 2 yeast data sets, and those 3 columns on the right are 3 tracks that you saw above. And you can see that they're fairly similar in terms of their RMSE, but for both of these data sets, the FRML is giving a lower RMSE, which is good, overall across all the phenotypes. And so we are encouraged that we are getting at least slightly better results than using FRs than with SNPs. We can also look at this as looking at the percent improvement in RMSE. And so we have yeast 100 on the top and yeast 200 on the bottom. Anytime you see a bar going up from 0, that means the FRs outperform SNPs. And if it goes down, then SNPs outperformed FRs. We're looking at just the 2 machine learning tracks. And so this is not considering GWAS here. In the first graph, you can see that nearly all of the phenotypes, which are what you have on your x-axis, outperform, or the prediction is outperformed using FRs versus with SNPs. The second graph, it is a little more evenly distributed, but the majority still have FRs outperforming SNPs, but there's still nearly as many with SNPs outperforming FRs. So we're not quite sure what the difference between these data sets are, but we're looking into it. We have played around a little bit with some of those parameters that I mentioned. And so you have the K values, the K-mer size on the top for each of those blocks within the larger block. And then your Y value is your kappa parameter. This is the one that allows for some insertions to occur in your FR path. And then the x-axis is your alpha parameter. And this is your penetrance parameter, which allows for a little bit of difference in the nodes that each of those individuals visit. So green is better. Those are the lower RMSE values compared to red. And you can see that the parameters really matter. Some parameters give you green, some parameters give you a lot of red. And then the other thing to notice here too is that you can't necessarily extrapolate from one data set to another in terms of which parameters will be best. They might be data set specific. We can also plot the phenotypes. Here's a couple of examples. The left one is from yeast 100, the right one from yeast 200. And so we have copper sulfate with the actual phenotypic value on the x-axis and the predicted phenotypic value on the y-axis. You want the actual predicted values to match, and so you're looking for a diagonal here. And you can see that the FRs-ML outperform the other 2 methods for the copper sulfate. And then also for 2% acetate, FRs-ML was the best performing method, but it still overlaps a little bit in its confidence interval with some of the other methods. And so in this case, it's not statistically significant. So in conclusion, we are using FRs as features for supervised learning models. And at this point, we are producing enhanced or equal performance compared to the SNPs. We're working to improve our prediction levels as well. So we are playing around with some of the parameters and improving our model in many different ways, including to increase its scalability, as well as to make it more general. So instead of just being able to work on compressed de Bruijn graphs, to be able to insert, to suck in other graph formats such as GFA. We're also looking at other interesting things to do, such as if we use both SNPs and FRs, would that improve our model? Or if we even use genes as features, would that help to improve our prediction? And finally, I'd like to just give a shout out to NSF and thank them for their support of this project. Thank you for your attention. I would be happy to answer any questions. If you have them, put them into the comments and I'll be happy to respond. Thank you.